When the motor neuron is stimulated, depolarization occurs and the sodium gate opens, causing sodium ions to enter the cell. Once the membrane potential increases, repolarization occurs and the sodium gates close. Potassium gates are then open and potassium ions rush out of the cell. There is now a high potassium concentration outside the cell and a high sodium concentration on the inside. Once the impulse reaches the synaptic bulb, the calcium gate opens and causes calcium to enter the bulb, to the cell membrane, and fuse to the cell membrane. Neurotransmitters, which are acetylcholine, are then released into the cleft and diffuse across the gap at the neuromuscular junction. They then bind to the receptor protein. Once the action potential travels along the sarcolemma, it goes down the transverse tubules that permeate the muscle cell and enters the sarcoplasmic reticulum, or SR. This causes calcium ions to be released. They bind to the troponin molecules on the actin, which is the thin filament of the muscle cell. This causes tropomyosin molecules to expose the binding sites for the myosin cross-bridge formation. This is myosin, which is the thick filament of the muscle cell. ATP binds to the myosin head and is then transformed into ADP and phosphate. This causes a cross-bridge to form between the myosin head and the actin filament. When ADP and phosphate are released, a sliding motion occurs as the myosin head changes shape so that actin is pulled towards the center of the sarcomere. When ATP binds to the myosin head once again, the cross-bridge ends and the process starts over. It transforms to ADP and phosphate, attaches to the actin binding site, and once removed, the sliding motion occurs. After contraction, relaxation occurs when acetylcholinesterase breaks down acetylcholine, so the motor neuron is no longer stimulated. Calcium ions then go back to the sarcoplasmic reticulum. ATP attaches itself to myosin, and the cross-bridge is reset. However, the troponin and tropomyosin molecules inhibit further interaction, as calcium can no longer bind to the actin filaments. Actin then returns to a relaxed state. And relaxation of the muscle cell occurs.